I think this is phenomenal. I mean, it's something strange going on. And of course, the etiology of this is either something affecting the way hemoglobin lets go of or cannot let go of oxygen, or it's affecting the way oxygen crosses through the microcirculation. Maybe there's no microcirculation, there's an impediment to microcirculation. Or maybe there's an internal mechanism of utilization. The cells just aren't utilized. I mean, there's lots of reasons, and the next slide goes over some of the factors that play into oxygen transport. Your acid-base status, if you're alkaline, you inhibit oxygen transfer. Most CFS patients have alkaline blood, if you measure it. Red cell 2,3-DPG level, red cell 2,3-DPG level. This is one of your compensatory mechanisms for living in altitude. So people living in Mexico City tend to have high 2,3-DPG levels, allows them to offload oxygen at low oxygen tensions at 10,000 feet. But that's lowered by citrate in this disease due to energy problems, and that can be affected. You could be phase lock loop to mitochondria itself. That is, it only pulls into the cell that which it can use, and if it can't use any, it doesn't pull it into the cell. There are a number of microcirculatory possibilities. Alkaline blood constricts blood vessels, impairing microcirculation. Low magnesium constricts blood vessels, impairing microcirculation. Effective low blood volume can be diminished due to volume contraction in a subset of patients. Low output, 3 to 5 liters per minute, that can impair microcirculation. And significantly so if you have concordant drop in vasomotor tone, and that will be particularly obvious in people with pain. Sex, it's been shown that women have a smaller cross-sectional cut of capillary cross-sectional area than men, significantly smaller. On that basis alone, something like this that affects microcirculation, and you have inherent in your sex significant reduction in capillary cross-section, you would be at greater risk for this. So women would be at greater risk for this, right? And are they at greater risk for this? The answer is yes. And you're looking at one of the biological parameters that would put you at greater risk. Not to mention the fact that women's cardiac output compared to a man is, on average, 10% lower in the first place. And their cross-sectional capillary area is significantly lower as well. Activity can make a difference. Female athletes, for example, on the basis of long years of heavy aerobic activity, have a wider cross-sectional cut than women who were never active as teenagers. So more sedentary women would be at particular risk for this. And those studies have been done by a guy in Canada who is looking at trying to reproduce fibromyalgia by interfering with sleep in a set of college volunteers, and he found that interfering with sleep could reproduce fibromyalgia, but men were resistant to it and athletic women were resistant to it. And you're looking, I believe, at the reason why. Mitochondrial function can be impaired by RNA cell activity, by xenobiotic poisoning from the gut especially, but other things as well. Heavy metals, especially mercury, growth hormone deficiency, discovered in this disease. DNA gene rearrangements or alterations of normal expression. And last of all, but perhaps most critically, reactive oxygen species, such as peroxynitrite. Now we're getting into Marty Paul's hypothesis. His hypothesis states really that, that the generation of peroxynitrite, which you see right here, occurs when nitric oxide is physically in the same location as superoxide. They will combine in a microsecond, a nanosecond, and produce peroxynitrite, which will only live for about a picosecond before it destroys something. It is the most deadly, the most powerful, and to some extent the most common serious free radical the body produces. There are other free radicals, but this one is really nasty. It's even nastier because it's produced by two reactive oxygen species, nitric oxide and superoxide, which are actually necessary for life, and you have a lot of it. You have a lot of these things. Fortunately, they're usually not in the same area. They're compartmentally kept apart. Superoxide is primarily produced in the mitochondria, except under certain conditions. And those conditions occur with ischemia, which is a consequence of excessive compensation for cardiomyopathy. And it's called ischemia-reperfusion syndrome. And accompanies loss of blood pressure and ischemic tissues due to loss of cardiac output, and a serious threat to organ function, I'll tell you. Pope John John Paul died of IR injury following sepsis. And the woman who had the feeding tube, Tereshaiba, essentially was put in that state through an IR injury. That's how bad IR injury. What's interesting about IR injury, ischemia-reperfusion syndrome, is that the primary organ that gets the most affected in IR injury and has the most trouble coming back is what organ? The brain. Superoxide is produced copiously in ischemic reperfusion injury outside the mitochondria and in direct proximity to nitric oxide, which is prominently produced in ischemia reperfusion injury, generating copious amounts of superoxide in ischemia reperfusion injury, which can be propagated from the locus all the way to the brain and injure it. And if it injures it good enough, you get to be Tereshaiba. If it injures it somewhat less than that, maybe you get to be a chronic fatigue syndrome patient without a brain. That's the kind of thing we're talking about here. How does the brain get affected by this? Marty Paul's hypothesis basically is that the mitochondria generates copious amounts of superoxide inside the mitochondria, where it is then enzymatically degraded to water, first by superoxide dismutase, then hydrogen peroxide, by glutathione peroxidase. It's degraded by SOD from superoxide to hydrogen peroxide, and then degraded by GPX from hydrogen peroxide to water. So basically the output of the mitochondria coming out is not superoxide, but water and CO2. That's normal. And if you can successfully, the point here is you generate tremendous amounts of energy, and see that ATP times 10 versus ATP in the glycolytic pathway. This is a copious amount of energy which will produce a copious amounts of superoxide, which if you can convert it all to water, doesn't put you at any risk. You get your cake and eat it too. You can make all kinds of energy and never have to suffer any problems from superoxide, provided you can take it down to water. If you can't take it down to water, then superoxide internally to the mitochondria can begin to cause oxidative injury to the mitochondria and bring the mitochondrial function down to the point that you can bring the rest of it to water. So what happens is you come down and then equilibrate eventually at a stable level of energy production so that you're no longer producing excessive amounts of superoxide, and there you will sit, not because someone had it in for you to give you low energy, but because by coming down to a low energy state, you become stable and will not further progress. So it's a very important idea. You equilibrate at a low energy state to save your life. But you have the consequences of that. Fascinating idea. This is Marty Paul. He's a professor of biochemistry at Washington State University. He's published some of his ideas and thinks the nitric oxide peroxynitrite mechanism is a common intracellular mechanism for the reproduction of the following diseases: chronic fatigue syndrome, post-traumatic stress disorder, fibromyalgia, and multiple chemical sensitivities. They're all linked together at the cellular level, and they're linked right here. The peroxynitrite hypothesis for human life is that in the presence of nitric oxide and superoxide necessary for life on this planet, unfortunately, under certain conditions, produce significant enough free radicals that you end up dying of this by slow oxidative death over a lifespan at best, 120 to 140 years of age. The point is that we use oxygen to make energy, we live with oxygen, and we will die with it. That's the idea. If you put an iron bar in an oxygen environment for 100 years and you return, it'll be a line of rust. If you put a human being in an oxygen environment and come back in 100 years, there'll be a line of dust. The bar got to rust in the same way that human beings go to dust. You go there by oxidative destruction, by the production of these waste products of oxygen metabolism, from which you get the very energy necessary for life. This is the free radical theory of aging, and I think it's a true one because there's no other way to explain all of this, except that's the way it is. We live in oxygen, we ultimately die by oxygen.